have more discussion with Dr. Manu Setty, and at the same time, I would encourage you to ask the questions in the middle of this talk. If you have anything, you can raise your hand. And also online, I don't know whether Zoom can hear me clearly, but if you have any questions during the talk, just feel free to raise your hand on Zoom and wait.
I'll just shout out your question. That's okay too. Yeah,
for sure. It is my pleasure to introduce my great collaborator, Dr. Manu Setty. Manu is an assistant professor in the Basic Science Division Computational Biology program in Public Health Science and a member of the PSLC. Dr. Manu Setty got his bachelor degree in computer science and engineering at the National Institute of Technology, Strasbourg, India. He got his master's degree in computer science at Columbia University and completed his PhD in computational biology at Cornell University and postdoc training at Sloan Kettering. His expertise lies in large-scale genomic data analysis with a focus on developing computational tools for single-cell and spatial data. Particularly, he studied gene regulatory networks and cell-to-cell communication mechanism. His lab is very productive in developing a lot of tools, including the Kompot, Mellon, Harmony, there are a lot. And today he will give us a great talk about his recent research, the computational model of differentiation and the disease trajectory. If you have any questions, just feel free to unmute yourself online or fill it at yeah, we have plenty of time today to do that. Okay, let's welcome Dr. Manu Setty for his talk.
Awesome. Thank you, Vicky, for the kind introduction. And it's I work in a steam plant building, but it's really nice to come across and meet with you all and share a bit about what we do in my group. As you saw, I changed the title a little bit. I wanted to just kind of broaden it to other things we are doing in the lab as well. So we come the way we are a computational biology lab, but we come from thinking about this specific biological questions. So we're interested in understanding how do cells differentiate. What is super cool, right? So we are complex organisms capable of abstract thought, but all our lives really began as one single cell, right? So 1 became 2, 2 became 4, and then through a cascade of changes, we are what we are in many ways, right? Then you have the idea that, yeah, that's fine. I'm gonna use the pointer. Great. You have stem cells that kind of go through a series of, let's say, gene expression changes and become different types of cells. So that's kind of the fundamental biological problem that we are interested in. And we are, so the goal is kind of how do we get at some general principles of how stem cell differentiation works. We are also interested, and as you can imagine, a lot of these processes tend to be disrupted in disease. So trying to really understand how these mechanisms are co-opted in tumorigenesis specifically, or just generally in metastasis too, for example. Okay, so that's kind of the sort of the biological motivation of where we come from, and then how do we do this, right? So we use, as Vicky mentioned, a lot of single cell and spatial data, always excited about new types of data and multimodal data. We work with a lot of collaborations. Every project, in a sense, we start as a collaboration, collect data, and build computational methods. And that's really kind of, let's say, the heart and soul of the lab is how do we use this data to solve this biological problem in collaboration by developing novel computational methods and computational tools that not only answer the question we are interested in, but can generalize to other systems for the people to use as well. Right. So of course, as you can imagine, this is like a massive problem. Obviously, we as a small group can't really solve all that. So we try to kind of come at it, break it down into 4 different angles, divided both by sort of biological motivation, but also the type of data that's available and our collaborators, and this kind of the 4 sort of arms of the lab. You can think of cellular dynamics. We are very interested in chromatin and gene regulation, also spatial organization, cell communication, but more recently really thinking about RNA processing, specifically of isoform composition as well. So what I'll do is, I mean, this is like a very large overview slide, but I wanted to give you a flavor of the different types of research we do in our group using some, I'll mostly spend our work in thinking about cell dynamics and talk about some of the algorithms we have developed and give a brief hint of the other things as well. And as Vicky said, please interrupt me at any point if you have any questions, any clarification. My goal is not to really go through all the slides put together, but hopefully have a good conversation to take away from. Cool. All right, so I'll dive in and I'll start about kind of what we started as a lab as well. And just to give a bit of perspective, I want to kind of start by thinking about, okay, how we think about data, how we think about problems, and kind of going back a few years now. This is my postdoctoral work, but I think it's a good perspective of how we approach research in my group. Okay, so kind of this is 2018, where single cell really was taking off, right? So we know single cell data was very powerful. There's been a huge number of algorithms that used basically single cell data, right? So essentially you can think of it as a bag of cells and you can put them in an order, right? So this is the trajectory or the pseudo time saying this cell progresses on to become these different terminal differentiated cell types. Right. So now that was interesting, and as data became more accessible, the challenge moved on to how do you start to model and think about these fit choices, right? So if we have one stem cell, it has to give rise to, let's say, 4 different differentiated cell types. There is a choice that the cell has to make at some point to become A or B, right? So that was thought to be very binary, right? A cell reaches some expression state, then it decides to become A or B, and then that was the idea of how these computational problems or methods approach this problem. But what the data suggested, not going too much into detail, is that this fate choice is not a binary choice, but something that's continuous. What that means, when a cell is kind of along its journey, right? So it does gradually acquire properties of becoming one cell type and lose property or ability to become other differentiated cell types, right? So thinking of this as a probabilistic choice, right? So for each cell, we think, or the data suggests, and there's sorry, this is kind of moving forward on its own. Experimental evidence also suggest it's not a binary choice, but there is this potential or probability of whether cell taking in one choice or the other. Therefore, we thought this is the idea that we need to think of how do we compute these lineage probabilities for each of these cells. Okay, so that was kind of the idea. That was the algorithm I developed and probably what got me this job as well. Thinking about cell-fate choices in a continuous way, thinking of cell-fate choice as something probabilistic itself. Okay, so now this is coming from a very data-driven observation. Now we need to build a formal method to actually explain this. Okay, so this is the idea we had. So nearest neighbor graphs, as I'm sure you're familiar, are very widely used in single cell data analysis because they kind of nicely follow along the manifold and in many ways proxy for manifold learning. The nice thing, I mean, of course, by definition, any graph you construct is an undirected graph, right? So what algorithms at that time already were very mature, like provided I know this is the start cell, you can essentially compute shortest path distances to say these are the pseudo time order, or this is the trajectory that an individual cell would take through to reach at this pseudo time. Okay, so now because we have this pseudo time, we can essentially transform this undirected graph into a bit more directed graph based on some approximate consistency with the pseudo time order itself. If A and B are neighbors, it's an undirected edge. It becomes an edge from A to B, a directed edge, if it's consistent with pseudo time progression. Okay, so now this is cool. That gives us some directionality, but the key idea that really allowed us to now get at the probabilities is to now thinking about these trajectories as a Markov chain itself, right? So we kind of started with the day observation about the data. We inferred these properties, and now we're finding the right kind of computational method or family of tools to start to understand these, to model these things. Cool. So that was the idea, and then yeah. So this is I think it's one, right. The nice thing is, and because of when we make sure that the assumptions we make are actually satisfied, and when we do that, we can automatically determine what are the different terminal states of the system. We're thinking about in a healthy differentiation state, and that gives us terminal states just based on properties of the stationary distribution of the Markov chain itself. Now with that, we can then essentially remove all the outgoing edges from these terminal states. That means any random walk anywhere will end up terminating in one of these terminal states. That then becomes really nicely an absorbing Markov chain. And that means by computing all possible random walks, we can actually get at these probabilities. So the cool thing here is now for each lineage or each cell, we now can compute what's the probability of that cell reaching in different terminal states. Okay, so for example, cell 1 here is at the start, that's kind of the stem cell, has high probability of reaching or differentiating to all these different terminal states. Whereas a cell here, for example, it's specified and committed to becoming branch A, therefore it has a single just kind of probability of 1, right? So there's no ability in healthy hematopoiesis to move to other lineages. And finally, I think the most interesting idea was states like this, which kind of maybe if we had thought of it as branching, we would think this is specified to become one fade, but this actually retains the ability to become other lineages as well. And this really allows us to now then quantify what's the differentiation potential, right? So it's like high potential, just computing the entropy of this multinomial distribution here. High potential means an ability to differentiate to many lineages. Low potential, it's functionally kind of set in what it's going to become. And it's really kind of reassuring because a lot of lineage tracing studies have kind of reinforced the idea that this is the observation that the like self-fed choice is something continuous, and it's just been very a valuable biological companion in inferring developing understanding of different biological systems. So that's kind of that sort of sets the stage. So I just wanted to pause and see if there were questions. I'm happy to clarify.
Yes. How informative this will be? For example, maybe this cell become a particular cell fate just because the nearby cell gave me the different, like two cell maybe identical, energy expression they may be the same. We'll decide its terminal signal. I don't know.
I think it's a fair point. In this case, we are the way this works, if there are two cells with identical expression profile, right? Let's say, then they would have identical probabilities here, because you're right. Like it's not the key point. The missing is also kind of what's the neighborhood of it, right? Here, this is strictly kind of cell autonomous or a property of the cell. So if there are two cells that are identical, here, at least with this assumption, they would have identical properties, but maybe by bringing in their actual physical space, one could then, that's a dimension, information missing, but bringing in that can help clarify that better. Yeah, does that answer?
Yeah. So there, I guess, potentially a way to quantify how informative, I guess, how informative this can infer the cell fate, like.
Yeah. So
maybe the distribution is uniform, then we cannot tell, or maybe the distribution is
correct. Correct. Yeah. So people have done is you can do lineage tracing experiments, right? So you can kind of set up. I mean, even something simple. You take a pool of cells, you put each cell with some kind of a different barcode, and then you differentiate in vitro, right? And then you do single cell sequencing, let's say even on a time course, right? So then you know each of those daughter cells, let's say, come from the same. Then you know as you trace along the cell division, that this cell just result in one cell type or multiple cell types, and it's shown that it is probabilistic. Where these assumptions are starting to fail a bit is whether the probabilities are become rigid here or here, right? I think that's some of the assumptions we built in maybe a bit more permissive, but there are actually people have updated our method itself to actually refine those probabilities. Kind of nice to see that. Yeah. Sorry, I go in and go. Yeah. Yeah.
May I interpret, for example, the A branch A, maybe the red blood cell?
Yeah, exactly. Absolutely, right? So you have, yeah, that's great. Sorry, I should have placed this in context a little better. The cell at the right at the top, you can think of this as like the hematopoietic stem cell, right? And then A is, yeah, as you said, erythroid. Maybe these are T and D because they're a bit more related for sure. So that's kind of the assumption that every cell is in many ways related to each other. That's for sure. That's something we are making in and going in here. And then that's kind of be actually true for the rest of the talk. I'm not going to be talking about discrete collections of cells today. We'll assume that all cells in many ways are part of one single trajectory for the entirety of the talk.
So I guess maybe it's not clear to me that how typically when you want to create a lineage cells, so you use some kind of, for example, some of the DNA structures. In the context of KKS, we're using the velocities in the evolution space. Do you have this kind of the data to allow you to construct the lineage, and then once you have the lineage, and then you can do whatever expression.
So the lineage itself is coming from gene expression, right? So it's like the way, I mean, you can think this is like a UMAP. Each cell is placed similar to each other in 2D if they are most similar in their high dimensional gene expression space. Sorry, I should have clarified. I mean, of course, we are taking all that information, shoving it down to 2D, but two cells that are closest here have the most similar gene expression profile, and that's what really defines the lineage itself. Now we are given the lineage, we are trying to kind of trace the history, right?
And then this is kind of what we use the sort of gene expression to create this sort of thing, and then you go back to look at the content and then try to look at which expression.
Yeah, so it's like, yeah, I mean, I think we use that as a state representation to build the state space. Then how are, let's say, the dynamic behavior happening, or what is the order of cells in the state space is what we are building towards.
But in the single cell, are you able to have the C sequence as well? So can you actually use those sequence to construct the lineage?
Right. I mean, that's an actual. I mean, with RNA, with this is all 3' end, so there's not a lot of SNP information. But there's efforts underway, especially using single cell chromatin like attack data. I mean, it's still incredibly sparse. In fact, Kelly Harris over at UW, she's working on something super cool. How do you recover this lineage phylogeny effectively, right? Based on single cell attack or single cell data. So very early stages, but I think efforts are underway. Yeah. Sorry.
So to be clear, you have a underlying Markov model that provides the structure. Are you estimating then all of the probabilities from in vitro experiments?
No, so the data is like an expression. Just for each cell, we have what is the expression profile of thousands of genes. Then we use that information to construct like a nearest neighbor graph, and then just inferring the probabilities. There's no additional experiments through which we are computing the probabilities, if that makes sense.
So intermediate state.
No, they're all observed. Every dot here is observed. So this is what the technology allows us. We can take like a aspirate from a bone marrow and profile thousands of cells, right? So that's one kind of the turning point of the technology, where previously you would take it and maybe generate like a vector of numbers. Now we can generate matrices of numbers because we have observation. Every dot we have an observation. Yeah.
Okay. So in the evolutionary models, they use hierarchical clustering to define or to identify this kind of lineage structure. What we gain by using this model?
Yeah, right. Right. So with the hierarchical model are things where here it actually now, I mean, I'll maybe talk about this a little bit, but we can now try to understand what are the specific gene expression profiles and genes that are changing, right? So there is potentially, like, for example, in immediate application to regenerative therapy. So one of the projects we're working on is looking at these kind of trajectories in the retina, right? So in the retina, there's like this X number of cell types. One cell type has been impossible to produce in vitro, right? So now if we can figure out what are the gene expression changes and factors that are controlling the changes that are triggering that. And that trigger is exactly this, right? Like where is the probability actually increasing to become that particular cell type? Now that allows then to like a practical or translational application. It's oftentimes these kind of transitions we see some of these tumors emerge too. So I think that's kind of where kind of thinking about a journey of a cell in many ways can actually help with this. Does that help? Yeah.
What's the difference in this case between the differentiation potential and like your pseudotime value, for example?
Right. I mean, I'm sorry, I don't have a plot. It's an inverse relationship, but it's not a perfect inverse relationship, right? Pseudotime.
You could say like a cell is somewhat similar to the root cell, but it's very committed, so its differential will be high, but it's pseudotime value.
Yeah. A stem cell will, by definition, will have low pseudotime and high differentiation potential, for sure, right? Because its potential is the entropy, so there's like high probability, right? So it's just a question of where pseudotime, in a sense, okay, if we assume that's just 1D axis, it's a one-dimensional representation. It doesn't tell you kind of the speed or where it's actually changing, etc. The differentiation potential can tell you where along the pseudotime is this commitment or specific, I mean, the probabilities are actually changing. So it's kind of complementary together, along with the probabilities, can give some kind of a description of the entire system. Yeah, does that help? Okay, great. Okay, no, that's great. Thanks for all the very interesting questions. And I think it's super helpful because also kind of sets a nice stage for the rest of what I wanted to talk. Yeah, so I think, so when I started the group here at Fred Hutch, we kind of took a step back and we have this nice graph representation. We can think about how maybe progression is happening, but we wanted to take a step back and really start thinking about how do we represent the single cell data itself. And then that's kind of work from what I'm going to talk about, which ended up resulting in this tool we called Mellon. All right. So when we think about differentiation, right, so that's kind of the cartoon on the left is an abstract version of a hill. Like you have a stem cell, it just kind of rolls down and ends up in these valleys, becomes different terminant differentiated cell types. And what I'm showing in the middle is just like a 2D version of it with a high resolution. Now, if we are able to measure all of these cells, one thing that's very interesting and important is to think about how are the cells kind of distributed along these valleys and hills and around this phenotypic landscape itself. If it was uniform, it would be like this, right? I mean, that's kind of definitional. But what biology means, well, one, the data already shows if we had this single cell data, but also there's fundamental reasons why there is the distribution of how cells are distributed along this phenotypic landscape, as we call, is actually not uniform, right? For example, as let's say this stem cell, it's ready, now it has to expand. You are generating a large number of cells which are very similar to each other, right? Or let's say there's apoptosis, then you're reducing a huge number of cells that are defining it. Sometimes there is response, like cells are waiting for a signal. As soon as I get a signal, they respond and move fast. So it really means that when we think of it, cell state density or some high dimensional representation of cell states and how cells are distributed along that space tends to be highly non-uniform. Okay, so this is kind of what so if you want to do anything about dynamics, if you want to think about how things are changing, etc., we need to get a good handle on how to represent this data, and that kind of was the challenge. Okay, I'm going to next kind of dive into how we actually do it, so I'm just happy to pause if there are any clarification questions about the idea. Okay, great. All right. So this is like, okay, so now we hopefully you're convinced that this density is variable. Then how do we compare cell state density, or how compute cell state density, right? Okay, so you can obviously, as you can imagine, densities can be approximated heuristics based on nearest neighbor graphs. So I mean, this is just now an example of a real data set of hematopoiesis. So the pink cells you see, these are the stem cells. They become monocytes. These cells are erythroid cells. Okay, for I think we don't need too much about the biology to kind of follow along today. All right, so if we just compute nearest neighbor graph or nearest neighbor distance and take the inverse, it's okay. I mean, I can tell because we understand the biology of it, this is not particularly great. The challenge is the data tends to be very high dimensional, and therefore one convenient way to do it is what I've been showing so far. You kind of compute the UMAP for a data set, compute the density in UMAP, and therefore that can be kind of maybe used as a proxy for cell density. Now this is tricky, right? Because if you slightly tweak the parameters, you can get fairly different looking UMAPs, and that's fine because these tools are designed to visualize the data. You're taking kind of this massive high dimensional data, shoving it down to 2D, and this kind of natural variation is not really surprising. Okay, so of course, this can lead to very different meaning or interpretation of the data if we stick to 2D UMAPs for densities. Okay, so then for the challenge is how do we estimate densities in high dimensional spaces, especially we are particularly interested in single cell phenotypic landscape manifolds that allows us to make certain assumptions about the data. And this work was led by Dominic Otto, who's a brilliant postdoc in my group. Cool. So this is the idea, right? Actually, before maybe actually I'm gonna skip past this. It should be okay. Great. So this is how we actually do this. Okay. So the key idea is we'll continue to leverage the relationship between nearest neighbor distance and density. So I'm gonna zoom in here, right? So I mean, just a simple thought experiment, right? If we have, well, I'm clearly in a sort of in this local area in a low density region because Wei is the person closest to me and he's a bit far away, but compared to you, you have a next neighbor sitting to you, right? So if you have high density region, the distance to your nearest neighbor tends to be small. And if you're in a low density, region, the distance to the nearest neighbor tends to be high, right? So that's the intuition we're gonna use. Okay, so then the cool thing is this can be, right, so for each cell or cell state, we can compute the distance to its nearest neighbor. And at this observation, of course, it's one data point, but can be described as this distribution called as the nearest neighbor distribution, which tells you what is given the data, what's the likelihood of seeing a neighbor in a given distance. Okay, that's quite nice. And there's a nice way to connect this distance distribution to the density. So that's kind of what's happening here. You have the distance to the nearest neighbor, and the density is a function of the distance to the nearest neighbor. And the nice thing is then we can start to share information across all of the neighboring cell states and compute something that is continuous and a continuous function. Okay.
It's not spatial resolve data, right?
Right, you're right. Sorry. This is, maybe I should not skip the slide. This is in the cell state space, not physical space. So imagine we take our single cell data, we get it down into like PCA, for example. We like to use diffusion maps, but we are in some kind of a state space. So it's, yeah, 20, 30 dimensions, not physical space. Yeah, sorry, yeah, right. Yeah, so just imagine this is all happening in like a 20, 30 dimensional space, which is a state representation of the biological data. Cool. Okay, so now this is really cool because what we now have, because we use this Gaussian process, is a differentiable density function. Okay, so what does that mean? So we essentially have a continuous density distribution that describes the entire phenotypic space of this, well, in this case, a differentiation process, right? Because all the cells are now related to each other. And this is really cool. And there's a lot of things we can do. And one of the things we can really do is, also do is evaluate what is the density of this space at single cell resolution. Cool. So, and just to give, why do we care? I mean, I want to give an example of the biology, but there's like a lot of exciting things we can do with this. And then I'm going to pause again for questions. Okay, so what we are now looking at is the same UMAP I just showed a few minutes ago of hematopoiesis. We have isolated cells based on the branch probabilities of just belonging to the B cell lineage, right? So the idea here is a stem cell, an individual cell take kind of journey through this state space to become a B cell. Okay, so that's pseudo time. So that's on the x-axis where each dot is a cell. and y-axis is the single cell density we have computed. Now, what you can kind of see, right? So like you have some peaks and some valleys. And the cool thing is when we kind of dig into the biology and understand what are these peaks and valleys, each of these high density regions is a known checkpoint along this B cell development. So especially for people who know about B cell development, you need to go through various stages to build a functional receptor. Each of these high density regions, essentially cells are accumulating, waiting for a signal or configuring something, and therefore, as they essentially wait in their gene expression profile, that shows up as a high density region because there's a lot of cell in a similar expression profile. Okay, and then the cool thing is low density region then of course are released from these checkpoints. That's where all the expression changes that define the next state are actually happening. And I'm not going into the detail today, but we have built like a gene change analysis. I mean, it's simple, but it tells you what is the gene that comes on first, what is the gene that comes on second, what is kind of the cascade of gene expression changes that occur here. And the really nice also thing is every high density peak I showed you is a checkpoint, but we also recover every known checkpoint, right? So we're really getting a nice complete description of how the system is behaving here. And that, in a sense, using this biology as some kind of a soft ground truth to make sure our system is working or method is working well. Cool. I just want to, I mean, it's more computationally heavy, but we have applied this to a whole bunch of biological system now, metastasis, mouse embryonic development, all kinds of biological systems here. And consistently what we observe is this, I mean, again, not surprising, but I think it's great that we have a formal way to think about this and then think about how this is breaking in disease, right? When we think of differentiation landscape, of course, this is kind of a very often used analogy, but what we really should be thinking about it, and we have evidence to show that, is cells are not just changing or distributed in a uniform way, right? You have a lot of cells which are very similar in expression profile, either waiting for a signal or waiting for some communication, etc. And once they receive it, they kind of very quickly change their transcriptional profile and hit the next kind of high density or a checkpoint state. And this kind of is a on and off sort of process where you can think of almost like a dam release, but kind of regulated. And that now our tool, Mellon, by quantifying this density at single cell resolution, allows us to start formally describing this. Great. Yeah, I'm gonna again just pause to see if there are questions about this. There's like a whole bunch of applications, but yeah, but anyway, I'll pause here.
So along the way, are you able to identify specific genes that basically will move their work from the stage to the stage?
Exactly, correct. We can, and I mean, I didn't show that here. I mean, one of the classic examples is when the stem cell takes on the identity of B cell. There is this transcription factor known as EBF1, early B cell factor 1, that's known to drive that change. How people have shown that if you knock out EBF1, you'll never get B cells. If you express EBF1 in, let's say, monocyte, it does become a B cell, right? So that's then through our gene change analysis, what we find, EBF1 is the top gene associated, for example, in the transition from the brown here, HSCs to the orange, which are these common lymphoid progenitors. So we can really describe not just with genes, but the waves of genes, right? So EBF1 comes first, and then this comes, and then this comes, etc. Yeah.
Yeah, I would imagine, like, for example, in here, it's like the hypothetical low density region also means like it's like a stable cell state and an unstable cell state.
Yeah, I mean, I think that's a very fair way to think of it, right? If you think of this like valleys and basins, this can be a basin where it's kind of held there, maybe waiting for a signal. I mean, it could be extraneous, or it could just be like, okay, I need this chromatin to open up and the enhancers to come together, then I can release, right? Yeah, it's yeah, I think it's a fair way to think of it for sure. But this also, I think the nice thing, I mean, I think that kind of thinking is not necessarily, I mean, not something we introduced, right? But now what we have is a way to describe and represent and identify those in across the system in high dimensions. Yeah. Yes.
those are
Yeah, this is all single cell RNA. We don't, but I mean, this is also multi-ohm data, so we have chromatin accessibility. Therefore, I mean, again, EBF1 is a prediction, and we can validate it based on prior knowledge. But we can then link, we can make a prediction of these are likely the transcription factors that are driving this change on the gene regulatory networks. I don't know, does that answer your question?
Both data translational and also, cellular status and kind of perfect biology explanation. I was wondering if you use transcription factor information or histological.
Yeah. Yeah, right. No, I think that's a great point. And if I come to it, one thing which, I mean, this is all just gene expression, right? And the chromatin accessibility is likely not behaving in the same way. Maybe the peaks are actually different, right? And that's exactly, well, I mean, I don't know if I'll get to it, but that's a project where we are showing chromatin and expression states are, of course, they're moving sort of together, but they're desynchronized, right? So chromatin opens first, that creates a bit more stable state in chromatin, and so on. So there is this, you can almost think of these like a chromatin density being a bit wider along this. And we have evidence to do that. That's, I think, yeah, that's another actual, yeah. But and histone modification is, again, such a great point. And one of my students' project is actually profiling single cell histone modification along with RNA in the same cell. Now we can do that for different marks in different experiments, use the RNA sort of as an anchor modality, right? Then essentially we have single cell by expression, chromatin, histone modification, and we can really try to build it all together. So that's also something where we're going towards. Yeah. Yeah.
How you differentiate the stem cell developed to be T cell and B cell? Understanding it's very hard step to differentiate that way either way. So is there already a solved problem or people want to use the data to better understand how?
Right. Now, I think that's a great problem. In this case, we're kind of completely ignoring the T cells. And the reason we are doing that is because this is all bone marrow, right? So this is all, the T cells actually develop in thymus, right? It's super interesting because a part of some of these, well, the idea is some of these cells exit after they have become, that's why it's called as a common lymphoid progenitor. They are out of their stem cell state, but they exit the bone marrow, migrate all the way to the thymus, and that's where the T cell development actually happens. So this is a small data set, but we have other data sets. I mean, this is, I don't know, like 5,000, but if you start putting 50,000 cells together, you start to see essentially a branch going off like this, a tiny, tiny branch, because those cells don't stay in the bone marrow, they're exiting. So you can start to really see the split between B and T. Not here because it's just too few cells. But because the development actually happens in the thymus, we don't see the T cell the differentiation represented here. Yeah. Any other questions? Awesome, great. So now I also wanted to talk about another method. Now, okay, this I told you how we can get a description and we can extract a lot of meaningful biology from the phenotypic landscape. What we can also do now, what we're super interested in trying to understand, if we have this healthy reference, how do we understand what are the changes, right, in a disrupted setting? Let's keep it simple and think of tumorigenesis. Okay, so this is the idea of Kompot. This is another method also developed by Dominic. Shows how awesome he is. So the idea is now if we have a phenotypic manifold in condition 1, condition 2, how do we compare? What are the states that are different? What are the expressions, gene expression profiles that are different? You can imagine any of it, right? Responder, non-responder, healthy, perturbed, healthy, disease, etc. So that's kind of the idea here. Okay, and it's of course not paired data, right? Yeah. Okay, so our Mellon density framework gives a natural way to actually do this, but do it in a very statistically rigorous way as well. Okay, so now imagine you have data from two conditions. Let's say we have that embedded again in some kind of cells, joint cell state space. Okay, maybe batch correction, etc. Okay, so the cool thing is now we can subset cells from each condition. The condition 1 is red, condition 2 is blue, and compute this continuous density function separately for each condition. Okay, now because it's a density function, it allows us to evaluate the evaluate density even in unobserved states, right? So therefore, what we can do using this function, now essentially trained only using cells from another one condition, but predicted density in the other condition too. Okay, hopefully that makes sense. And then from there, because we have single cell for each cell, what is the predicted density in one condition, what is the predicted density in another condition, we can actually get a fold change that actually gives us measure of the effect size of differential abundance. Okay, so for example, oh, I should use this. Okay, so here you see this largely cells from the blue condition, and here you see, okay, here largely cells from the red condition. And by computing this fold change, now this shows there's higher abundance of cells from the red condition here, whereas higher abundance of cells from the blue condition here. So that's the idea. Okay, any quick questions? I mean, this is just, of course, effect size, but yeah. Cool. So, I mean, this is really nice because, I mean, this was, of course, this part was straightforward, but what we can get at now is this volcano plots, right? So Dominic, and going a little bit in a bit later, but he worked extensively to kind of get at uncertainty measures. And when you have uncertainty measures, you can start to build these confidence measures, and you can get a posterior trail probability for at single cell resolution. I think this is the first time where you can actually do this kind of differential abundance analysis at single cell resolution with a formal P or a posterior tail probability and also a fold change. And there's ways to do when you have multiple samples from the same conditions as well. Okay, so I mean, okay, this is a bit busy, but let me walk you through. Okay, let's start with panel A. So this is again hematopoiesis, but this is in the mouse. Okay, so murine hematopoiesis. This is quite a few cells here, but this is a study that's looking at aging in hematopoiesis. Okay, we have cells from the young mice, cells from mid mice, and cells from old mice. It's a collaboration with Stanley Lee here in HB and TST. Cool. So now we can do pairwise comparisons, young to mid, young to old, and mid to old. Let's start with young to mid, right? What's interesting, so this gives us the fold change, but this also now tells us which is actually it gives you a way to threshold them and say these are truly significantly differentially abundant. I mean, not super obvious from here. What's interesting is when you go from young to mid, the main changes tend to be in the kind of the more mature populations. Whereas when we go from mid to old, there's an increasing accumulation of this stem cell population. Now, this is interesting, and it's well known. As a function of age, stem cells become a bit more dysfunctional, where rather than differentiating, they kind of tend to hang out and be in the stem cell state itself. And that actually leads to reduced numbers of immune cells as you age, right? And in fact, after some age, there's just very few B cells left in the bone marrow. And I mean, this is maybe perhaps a lot of things that existing tools could do. But what we can do now is actually, because we have single cell resolution, we can really compare what's the pseudo time, etc. And I'm sorry, it's not super visible here, but it's in many ways what this data suggests is the stems, with age, stem cell are becoming more STEMmy, right? So they're kind of reinforcing the gene expression program that defines what a stem cell is rather than trying to differentiate, which I think is potentially very interesting. Cool. So this is, I'll go through this and again, maybe pause for questions. This is differential abundance, right? So like, what are the states that are changing? But we also wanted to look at differential expression. What are the genes that are changing between these conditions across the data? Okay, so this is kind of the idea of how we're doing this. I just want to kind of just to make sure we are on the same page, we are not looking at differential expression between two groups, two conditions or two clusters of cells or two different groups. We are looking at, let's say, if this is hematopoietic stem cell, this is young and this is old, what is the change in the same compartment across the two conditions we are looking at? Okay, so that's a different use case than what is the difference between, let's say, T cells and B cells. It's saying what is the difference within T cells between, I don't know, treatment versus immunotherapy or whatever it is. Okay, cool. So this is again like a bit of a technical detail of how we do this. Now each dot again is a cell. Think of this as some kind of a pseudo time axis, and this is, let's say, an important gene that defines this axis. I mean, this is actually representative of the single cell data, right? Because this, I mean, in a sense, for each, in the data set, we are maybe capturing 5 to 10% of the transcript. So there's massive zeros and massive dropouts. So it's a very sparse and noisy data, and people who work with it are super familiar with this, right? But what we can do, of course, that's noise. We can impute, right? So what we do is essentially fit a Gaussian process that fits a smooth, assuming that things are changing smooth. That's an assumption we make, and I feel confident about that. We share information between cells, and therefore we can get a smooth imputation across them. And the cool thing is we can do it separately for each condition, right? So we use cells and expression of that specific conditions and fit two functions. So we can fit a smooth function mapping the state space to the gene expression across separately for each condition. There we computed density. Here we are just computing a function to build expression mapping for each condition. Yeah. And because it's continuous, now we can evaluate the predicted expression in the other condition. Now, again, similar story, it gives us now a fold change at single cell resolution for each gene and each cell between the two conditions. Okay, of course, I mean, that's how would be a lot of information. So how do we assess the significance of it? And Dominic came up with this, what is a really cool idea. The idea is you have imputed expression. We can get at the uncertainty of this imputed expression. This took a lot of work as well. What that means is we have for each cell, what is kind of the uncertainty, right? Or the standard deviation of the expression. But because of the way the algorithm is built, you actually have a measure of covariance too, right? So now what we have is how does this particular gene covary across the entirety of the state space. Now we can do this for the fold change too. So rather than thinking of whether the change has a difference in effect size, we can now try to say whether a gene has significant difference in its covariance pattern between the two conditions. Okay, so that can be measured by this metric called as the Mahalanobis distance. I know there was a lot of information, but I want to just walk you through an example, and that hopefully clarifies things a bit better. Okay, so this also allows us to then do this kind of pseudo quasi volcano plots where x-axis is the mean fold change, y-axis is this metric that tells you whether this gene has significant covariance difference between the two conditions. And why we did this hopefully becomes informative in the next slide. Okay, so we're looking at the same murine hematopoiesis data set. Again, different cell types, it's kind of matters less. We're looking between old and young cells. And then each dot here is a gene. Y-axis is, and we are using all the cells here across all cell types. We are not even looking at one cell type. So I think with this, so we are looking at all the cells here effectively. Each dot here is a gene. Y-axis is the significance Mahalanobis distance metric. Okay, so we'll start with the gene here, top right, very high positive fold change. That means it's higher in the older cells compared to young cells. And you can kind of see it, right? For the most part, maybe except for this group of cells, this is the average across all cells. This plot is the same UMAP showing the expression change of this gene in each cell across between old and young. So kind of consistently, this is a gene that is upregulated as a function of age. I mean, again, not surprising, this is one of the HLA genes. It's known to be something that gets upregulated as a function of age. Okay, Right. So on the other end, you have this DIN LL1, and you can kind of see mostly again downregulated, which means the average log fold change is low. But what's really cool, and it's very unique to the kind of thing, the approach we have built, is that you have gene that is kind of zero, like almost no fold change across the population of cells, right? But it is a gene that changes in a significant way in terms of its covariance patterns between the two conditions. Okay, so that's kind of reflected here. You have this gene really upregulated or downregulated as a function of age in this cell type, but upregulated as a function of age in this cell type, and maybe some downregulation here. Together, the effect size is low, but it's a very strong covariant pattern. Now, this is really cool because rather than kind of saying, okay, I'm going to look at this cell type and look at these genes, you can start with the entirety of the data set, identify what are the genes that are actually different, and then kind of try to get at the biology of it. And one thing I don't have slides here. Two interesting observations we have made. Oftentimes, there is a particular pathway that's impacted. For example, even in aging, this pathway of antioxidation, genes involved in antioxidation are actually impacted as a function of age, but different genes change in different direction as a function of age, which we would actually miss with traditional analysis, but we can pick this up here. We have also seen cases where, for example, metabolic changes go up in epithelial cells, but maybe potentially as a compensatory mechanism, the same genes are sort of downregulated in T cells in the intestinal epithelium, just even in like, I think it's like, I forgot the condition, but anyway. So I think those kind of opposing but structured changes is something we can start to pick up with this, and also generally global changes that are happening across different cell types. So this has become, I mean, it's recent, but we are actively using this across a whole variety of projects to kind of come from of a perspective of differential expression from a very different view, rather than trying to think of cell types, especially when you have something that's very continuous and a system that describes as an entirety. You can start from the system and then try to identify what the genes and then interpret. So yeah, that's the quick summary of Kompot. Again, happy to pause and take questions on this as well.
I clarify how you by covariance, because you have data from just two conditions and you have different cells in like one gene. So what is the covariance?
Right. Sorry. So for it's the covariance of the gene across the different cells. Because right, so you can get a measure because of the Gaussian process. You can get an uncertainty for each cell and a gene, but you can also say how does that specific gene covary between any pair of cells, and we can do it across all pairs of cells. That gives you the covariance matrix per gene per condition. Then you have two matrices because we have two conditions. Then you can measure how different are they using the Mahalanobis distance. Does that help? Okay, thanks. And that really is what allows us to now, in this group of cells, this is going up. In this group of cells, this is going down. Or let's say it's in one condition, in this group of cells, it's high. In the other condition, it's not. But maybe if we look at this particular cell type, maybe it's tiny, then we wouldn't detect it, right? Or if there is opposing changes, if we take the mean, it would cancel out. But because there is strong covariance pattern in one condition that's gone in another condition, now the gene ranks high. So that is where this is the type of that helps find new biology potentially, right? Yeah. Mm-hmm. Any more questions? Okay, I'm gonna like maybe just because we have some time, just talk a little bit of the chromatin angle we're thinking about that Vicky asked a question about, and thinking about when we have multi-omics. The cool thing about with these single cell technologies is when we have well, I'll come to that. Okay, so the kind of what is the premise of this, right? So we know that gene expression is regulated by chromatin, right? So you have chromatin accessibility. That means there are transcription factors or whatever other elements bind to the chromatin and then open up and lead to the expression of the genes. So chromatin turns on RNA, RNA of course then leads to protein, which then can go bind chromatin again. Okay. So one of the really important and interesting things is they're not kind of moving in together, right? So there's always like a delay and a lag. So a change in chromatin might not immediately reflect a change in expression, right? And then this is work led by Connor Finkbeiner, who's a grad student in Mellon. So our goal, now with single cell data, which is super cool, but then with single cell multiomics, where from each cell you can get both its gene expression profile and chromatin profile. So we have paired information now allows us to really start to investigate how this is happening, or what is actually happening. Okay, so yeah, so I mean, we're going to talk today about a bit of the retina. So top is RNA, bottom is attack. So these are separate UMAPs that were generated separately for RNA and separately for attack. But then now we have added a connection between the RNA and attack to know which cells the same came from. Okay. So, and just to give a bit more of a premise, I'm going to just talk a bit of the biology and maybe skip over the technical details for this section. Let's look at this example. Now, this is the expression of OTX2. OTX2 is a gene that is a known master regulator of cone fate, right? So you have expression of OTX2 here that leads to this fate choice, and that cells become cones. Okay. Now, let's look at the locus, right? So this is like a genome browser, a track of accessibility, where we did pseudo bulk for each of these cell types and looked at what are the loci or the regulatory elements that regulate the expression of OTX2. And in specifically interesting is DHS4, which is a known master, like a key regulator. So if you take out DHX4, there's no cone cells that are developed in the system. Okay. So we know that regulates. And as you can imagine, if it's a cone cell, because in cones, you have all these accessible elements, like there is some protein or transcription factor binding to the DNA and keeping the chromatin open that allows you to get this readout. Okay. Now, what's interesting, if you look at, for example, the cycling precursors, which are just one step before the cones. Okay. What you see is, what's interesting is DHS4 is already accessible. So there was something bound in cycling precursors that is keeping the locus open, but the gene expression is not turned on, right? That gene expression turned on means that the cycling precursor, which still retains ability to become other lineages, is going to commit and become a cone cell. So this is what's called as kind of priming. Again, we didn't discover this at all. It's actually well known in this field, or just generally the idea of enhancer priming is actually very well established. Now, on the contrary, if we look at kind of other terminal lineages like radial glial cells and horizontal cells, as you can imagine, this kind of completely wiped out, right? Whatever is bound in cycling precursor has been kicked off. Therefore, that's kind of completely wiped out. But what's interesting is if you look at these precursors, it actually still retains some degree of openness. Okay, so oh, I'm sorry. I know it's still. I just want to don't hold back people if they have to have schedule at one. Yeah. Yeah. Sorry. Yeah. So I think with the RGC precursors, it's still kind of kept open, right? I mean, that's interesting, right? It's kind of crazy. Like cells. These are cells that are likely not ever going to be become RGCs, but they still have this locus to some extent open. So this is kind of the idea of the plasticity. So then the question is, how do we actually do this in a systematic way? I'm going to just go through this fast so that I don't hold back too much. So the idea of density also allows us to do this in a systematic way, right? If your expression, let's say in this case, chromatin changes before, so chromatin changing first, therefore you'll have a much lower density at the chromatin level, whereas you'll have a higher density at the expression. So that's the idea we use. That's, I think, and what's striking is we see very large differences across the entirety of the differentiation landscape. There's very few cells that are basically identical. There's always one changing before the other. We have a way to associate what are the features and the genes and the loci that are associated with this. And we can kind of really go in and I'm going to skip past the whole thing, interpret all of this. And just kind of at the summary stage, what we started thinking about, okay, how are the cells distributed? The distribution tends to be different when you look at different modalities, and using what are the features associated with this difference, we can actually figure out an entire gene expression program and the regulatory network that allows these cells to achieve this. And this is an example of the gene expression dynamics I mentioned earlier. Cool. So I think that's kind of, and then we're kind of taking a lot of angles, thinking about single cell communication and spatial organization, and also thinking about how do we use long read single cell RNA-seq to inform isoform dynamics. So that's kind of, yeah, that's all the work in the lab. Huge thanks to all the group members who really are the heart and soul of the lab. Our collaborators, funding source, and really it's such a great pleasure to come talk here, and thanks so much for your time and attention as well.